Hi, I'm Dr. Pugh. I'm a researcher and pathologist based at the University of Birmingham. My field of interest is the immune control of viruses in the context of inflammatory and neoplastic diseases, and I'm especially interested how the immune system controls viruses in the spatial context. Our aim is essentially to understand the immunological and the immunopathological basis of inflammatory and neoplastic conditions. My focus in particular is on how the immune system does this within tissues. I think the spatial component will become more and more important. I mean, previously we've disaggregated tissues to understand what lymphocytes and other immune cells are doing within tissue. I think going forward we'll keep tissues whole and try to understand the role of the immune system in a spatial context. I think it's really important to understand when markers are co-expressed in any given cell and to understand the spatial relationships of those cells to each other. What high multiplexing enables you to do is to really assess in a qualitative and quantitative fashion when markers are co-expressed and how those cells relate to each other. And that's really important in understanding the biology. In terms of data analysis, as well as the qualitative analysis that I would do as a pathologist, we've also done a number of quantitative analyses. This usually starts with cell segmentation, followed by phenotyping, and then statistical tests. So we're able to enumerate, for example, the number of cells or cell phenotypes that are present, but also look at the spatial relationships of those cells in a quantitative fashion. Okay, our main research goals for using the COMET was to understand a quite complex immunopathological phenomena within placental tissue. The cells within the tissue were of multiple cell lineages, and therefore we really needed a multiplex platform to help understand the biology and the pathology of the disease that we were studying. I mean, I would advise the COMET to other researchers and clinicians who are performing research on tissue. It's particularly useful in the studying immune infiltrates, but if you want to study the expression of any type of marker within tissue, then the COMET's an ideal platform. I find COMET user-friendly. There's a user-friendly software interface. The machine itself is very intuitive to use, but also there's enough flexibility within the software that enables you to adjust conditions if necessary to. Going forward, I'll be using COMET in my future projects to understand the role of the immune system in controlling viruses, and looking forward to using this technology in the context of virus-driven cancers, and hopefully that will provide insights into new therapeutic approaches and deepen our understanding of the disease. My main advice to researchers who are wishing to get into multiplexing is to ensure that there's robust validation. The COMET is particularly useful for this, as you can use antibodies that you've already validated. Moreover, there are well-defined validation protocols built into the system, which enables whole panels to be worked up within a number of weeks.